In the second part of our series on adaptive immunity, we're going to discuss how lymphocytes recognize their antigens. The types of antigens that are recognized by B cells and T cells are distinct. B cell receptors can bind soluble, free-floating, or surface antigens. These can include proteins or pathogen surfaces, lipids, carbohydrates, or nucleic acids, or really anything that can bind to this B cell receptor. T cells, on the other hand, only bind to small peptide antigens. Peptides are strings of amino acids, usually about 8 to 20 amino acids long, and these are presented on the surface of antigen-presenting cells that express a complex of molecules called the major histocompatibility complex. T cell receptors can only induce T cell activation when they bind to a peptide being presented by MHC. Going back to B cells, naive B cells express their antigen receptor only on the cell surface. And the isotypes that are expressed here are IgM and IgD. Naive B cells do not secrete antibodies. But when they become activated, these antigen receptors can also be secreted as antibodies. And they can be secreted as one of 4 primary isotypes of antibodies: IgM, IgG, IgA, and IgE. IgM is the first isotype secreted by activated B cells. But B cells, once they become activated, can switch their constant reagents and start secreting a different isotype. This is a process called isotype class switching, which we will discuss later. A single B cell only secretes a single isotype at a time. T cell antigens, on the other hand, require presentation of a small peptide by MHC class 2, as previously discussed, or MHC. There are 2 classes of MHC, MHC class 1 and MHC class 2. MHC class 1 is expressed by all nucleated cells. MHC class 2 is expressed only by a subset of cells called professional antigen-presenting cells, and those are primarily dendritic cells, macrophages, and B cells. MHC class 2 molecules bind to T cell receptors expressed by CD4 positive T cells. MHC class 1 molecules bind to T cell receptors expressed by CD8 positive T cells. MHC class 1 specializes in presenting peptides from cytosolic antigens, and that is why MHC class 1 antigens generally are derived from viruses, although certainly not always, and are specialized in activating CD8 positive T cells that have antiviral functions. MHC class 2 presents antigens that are extracellular and that have been phagocytosed. This includes antigens that are in phagolysosomes or antigens that are taken up by other means of endocytosis. In humans, the major histocompatibility complex is encoded as human leukocyte antigens. So if you see the term HLA, or human leukocyte antigen, that is referring to MHC. The MHC genes are on the same chromosome and tightly linked together. There are 3 MHC class 1 genes, A, B, and C, and 3 MHC class 2 genes, DP, DQ, and DR. These MHCs each bind a range of peptides based on their biochemical characteristics. This binding to peptides is non-covalent, but it is pretty stable. And under non-infectious conditions, MHC molecules bind self-peptides. Under infectious conditions, MHC can bind to foreign peptides derived from pathogens and present them to T cells. There are 6 unique MHC genes that we just discussed, 3 class 1 genes and 3 class 2 genes. And so this increases the diversity of peptides that can be presented by any individual. But MHC is also the most polymorphic set of genes in the genome. And that means that there are a number of MHC alleles within a community, which increases the diversity of peptides that can be presented within a community or population. MHC genes are expressed codominantly. So here, for example, I've shown, let's call this an epithelial cell, and it has 2 copies or 2 alleles of MHC HLA-A, 2 alleles of HLA-B, and 2 alleles of HLA-C. The polymorphism of each of these alleles is designated with a number. And so here we see HLA-A2 or HLA-A20 as a designation for the particular allele that is being expressed by this cell. Antigen-presenting cells additionally present MHC class II molecules. And so they have up to 12 different MHC molecules presented on their surface.